Today we are going to learn about TCA cycle, another major pathway in the metabolism of carbohydrate. In the previous video we have learned about glycolysis, the steps, what are the regulation, why glycolysis is needed, and come to today's topic. This is TCA cycle. So at the end of glycolysis we have got pyruvate. The pyruvate again converted into the acetyl CoA with the help of pyruvate dehydrogenase. This acetyl CoA enters into the TCA cycle. TCA cycle, we can call it the final common oxidative pathway. Citric acid cycle may be considered as the final common oxidative pathway for all foodstuffs. All the glucose, fats, amino acids coming in the TCA cycle. All the major ingredients of foodstuffs are finally oxidized through the TCA cycle. Almost all the biochemical processes use ATP for meeting energy needs, that is for muscle contraction, for active transport, for biosynthetic reaction, etc. So where the TCA cycle is going on? TCA cycle is going on in the matrix of the mitochondria. Now come to the steps of TCA cycle, or you can say the reactions going on in the TCA cycle. So this is the whole cycle. We can start from here. That acetyl CoA. Acetyl CoA enters into the cycle and oxaloacetate is going on. They both are producing the citric acid. First step of TCA cycle is formation of citrate. Here the enzyme acts citrate synthase. So with the help of citrate synthase enzyme, acetyl CoA and oxaloacetate is producing the citric acid. That citrate is converting into cis aconitate with the help of enzyme aconitase. That cis aconitate is converting into the isocitrate. Isocitrate then converting into the oxalosuccinate. Here one NAD plus molecule is converting into NADH plus H plus. Here the enzyme is isocitrate dehydrogenase. And when one carbon dioxide molecule is going on, the oxalosuccinate is converting into the alpha ketoglutarate. That alpha ketoglutarate is converting now succinyl CoA. Here another NAD plus molecule is converting into the NADH plus H plus, and another carbon dioxide molecule is coming out. This succinyl CoA is converting into succinate. Here GDP is converting into GTP. Here the enzyme is succinate thiokinase. Succinate is converting into fumarate. Here FAD is converting into FADH2. Fumarate is converting into mutase. Here the enzyme is fumarase and malate, fumarate to malate, and malate is last converting into the again oxaloacetate. Here NAD plus molecule is converting into NADH plus H plus, and the enzyme is malate dehydrogenase. So this cycle you have to remember what is producing, and when the conversion is going on, what enzyme is activating. So if you can appreciate, you can see that where the NAD plus is converting into the NADH plus H plus, that enzyme is always dehydrogenase enzyme. Here, when isocitrate is converting into the oxalosuccinate, NADH molecule is producing, and the enzyme is isocitrate dehydrogenase. Here you can see another, when alpha ketoglutarate is converting into the succinyl CoA, enzyme is alpha ketoglutarate dehydrogenase. Here, when FAD is converting into FADH2, enzyme is succinate dehydrogenase. And when malate is converting into oxaloacetate, enzyme is malate dehydrogenase. You can make your own numerics to remember this cycle, but you have to remember it. Now if we check the ATP generation in the one cycle of the TCA cycle, here when isocitrate is converting into oxalosuccinate, in this step one NADH plus H plus molecule is producing. So from here we can get 2.5 ATP. Another step is here when alpha ketoglutarate is converting into succinyl CoA, another 2.5. Then here already ATP is producing in the form of GTP, that FADH2 in the process succinate to fumarate. FADH2, from FADH2 we can get 1.5 ATP, and here another NADH plus that is going in the that give us 2.5 ATPs. So from step number 3, means isocitrate to oxalosuccinate, we are getting 2.5 ATPs. From alpha ketoglutarate to succinyl CoA, that is step number 4, we are getting 2.5 ATPs. From succinyl CoA to succinate, we are getting GTP in the form of ATP in the form of GTP. So 1 ATP we are getting here. Then from succinate to fumarate, step number 6, succinate to fumarate, FADH2 is producing. So from 1 FADH2 we are getting 1.5 ATPs. And in this step, malate to oxalosuccinate, step number 8, we are getting 2.5 ATPs. So from there we are getting 10 ATPs in one cycle. Now come to the regulation. Again, when the hormonal regulation comes, two hormones are very important, insulin and glucagon. Insulin promotes the TCA cycle and glucagon inhibits. Another, if you check it, ADP is an important factor for the regulation of TCA cycle. TCA cycle is known as amphibolic pathway because both the catabolic and the anabolic, both pathways going on here. TCA cycle is both catabolic and anabolic, so it is known as amphibolic pathway. So that is all for TCA cycle. Nothing much you have to remember. ATP generation you have to remember, and the steps where which enzyme is going on, that you have to remember. That is it for today. Thanks for watching.